Hello friends, today we are going to discuss about natural selection, one of the important bases of neo-Darwinism. We have already discussed neo-Darwinism. You can refer my video, the link is given in the description box. So first of all, we will see what is selection. It is a combination of all forces that cause differential survival and differential reproduction among genetic variants or among the individuals that are genetically different. It is of two types, artificial selection and natural selection. Artificial selection, here the selective agencies or the factors of selection are not by chance or by the nature, but it is of human choice. In natural selection, these selective agencies are not of human choice, but these are natural. So what we can tell about natural selection? It is the machine that drives evolution, or in other words, evolution is driven by natural selection. Natural selection results in descent with modification. That is, or it means, those features of organisms of a population are favored that makes them better adapted to their environment, or those are responsible for the better adaptation of any individual or organism to their environment. So let us understand natural selection with this flow chart. It is differential contribution of the alleles of a gene to the gene pool of a population in the next generation. It helps in the differential contribution of only those alleles of the gene to the gene pool of a population in the subsequent generation, which is responsible for their better adaptation. Nature of natural selection. First is differential reproduction. It means individuals better adapted to the environment reproduce at a higher rate, that is, produce more offspring compared to the less adapted ones. Hence, better adapted individuals contribute greater percentage of genes to the gene pool of the subsequent generation compared to the less adapted ones. And this brings evolutionary changes because there is the change in the gene frequency by the differential reproduction, or because differential reproduction of genes or gene combinations are favored. This differential reproduction is achieved by increased adaptability of the individual to the particular environment, decrease vulnerability to the factors which are responsible for the death of the individual or organism. Better chances of sexual selection, that is, these have the more chances of being sexually selected. Positive selection of useful genetic variability. Useful genetic variability are positively selected and then these are contributed to the subsequent generation. Now let us understand this differential reproduction. With the help of this figure, this is the initial population in a particular environment. Here this is the wild type and this is the mutant type. Wild type are due to the wild type genes and mutant are due to the presence of mutant genes. Now, due to some factors, there is the change in the environment, and the change is such that mutant varieties are better suited or better adapted to that change environment compared to the wild type. Now, the mutant ones survive and reproduce more often compared to the wild type, and there is heredity. That is, surviving mutants produce more mutants. They have the reproductive advantage And this is known as differential reproduction because there is difference in the reproduction of mutant and the wild type. Mutant one reproduce more often compared to the wild type. Hence, differential reproduction. So, what will be the end result? Mutants are now more common in the population. Because of the differential reproduction, their number in the population increases compared to the wild type. And we already know that these mutant varieties are due to the mutated genes. So what happens? Because these mutant varieties are advantageous over the wild type, so the mutated genes that are responsible for these mutant or these traits keep on increasing in the population over a long period, and this results in evolution. Second is encouragement of beneficial genes. Natural selection encourages genes that results in highest adaptive efficiency between population and its environment. That means it encourages those genes which makes the population better adapted to its environment. Reproduction and survival of more suited individual is favored in each generation. This brings improved adaptive relation between organisms and environment in subsequent generation, and hence organism becomes better adapted to the environment generation after generation. So we can say the natural selection results in evolution, or in other words, natural selection is a creative force in evolution and it favors only those gene combination which makes an organism better adapted to the environment or better suited to the environment. We are going to discuss about modes or types of natural selection. We already know that natural selection is differential survival and differential reproduction. Differential survival means only the well-adapted individuals to the particular environment survive. Differential reproduction takes place of those adaptations that are genetically controlled and heritable, because these only can pass from generation to generation. In other words, natural selection is differential reproduction of alleles which help the survival of next generation. It produces evolutionary change because it acts on the variability within a population that is heritable. Now the modes of natural selection. Let us see them one by one. First is directional selection. Directional selection is change within a population in one direction in response to the change in the environment in a particular direction. Individuals that change in response to environmental change and become most adapted are favored. It means that individual which changes in response to environmental change and become adapted to that environment are favored or are selected, or these survive. So it is progressive selection. It occurs during or after the environmental change. Salient features. Occurs due to change in environment in the particular direction. Non-average or specialized phenotypes are favored and normal or average individuals are eliminated. It alters the mean value of the trait. If we represent it with the graph, this shows a bell-shaped curve. Here before the selection, the graph is like this, and after the selection, it has shifted towards this direction. So individual towards one end, these are eliminated and these are added towards the other end. Also the graph has shifted from here to here. Hence the mean value has also shifted from here to here. So we can say that it alters the mean value of the trait, and the population shifts in one direction. Favors the accumulation of mutations that increases fitness in the changing environment. Because the environment is changing and hence the population or the individual should also change. So it favors the accumulation of the mutation that increases their fitness in the changing environment. That means it results in the change in the population in order to get adapted to the changing environment. Normal or average individuals are eliminated because individuals that are extremes are favored. It brings about progressive evolution. We have already seen that it shows the shift in the population. Example, industrial melanism. Earlier, before the selection, in the population there was more frequency of light colored moth. With the industrial revolution, the population shifted and now there is more frequency of dark colored moth in the population. Second is resistance of insects to DDT. Initially in the population they were mostly DDT sensitive insects. With the continuous use of DDT for few generations, the populations shifted to DDT resistant. Now there are more frequency of insects that are DDT resistant in the population. Now second is stabilizing selection. It is also known as normal selection or centripetal selection. It occurs when there is absence of large scale environmental changes for long period, or in other words it takes place in the unchanging environment or the environment which is almost constant. Environment is constant, so mutations are eliminated. Since the environment is constant, so genetic variation is not required, so mutations are also eliminated. Keeps a population genetically constant. Hence, in the absence of the mutation, it keeps the population genetically constant. Eliminates extreme variance. Also, average or normal phenotypes are favored. So, the normal phenotypes are favored and extreme phenotypes are eliminated. Frequency distribution appears exactly as earlier before selection. So, the distribution of frequency appears same before and after selection. Now the salient features: operates in unchanging environment. It takes place in the environment which is almost constant. Introduce homozygosity in the population. Since the environment is almost constant and hence homozygosity in the population is introduced. Average individuals are favored. So it favors the average individuals, and extreme phenotypes or individuals are eliminated. Checks accumulation of mutations. Since environment is unchanging, so accumulation of mutation is also checked or it is eliminated. Tends to arrest variance and evolutionary change. Because the environment is constant and average individuals are favored, hence it tends to arrest variance or genetic variation and also arrest evolutionary change. It is rare because the environment keeps on changing, as it is not possible that the environment remains constant or it doesn't change, and hence this type of the stabilizing selection is also rare. If we see this graph, this is bell shaped and it is before stabilizing selection. These are extreme phenotypes and this is the intermediate phenotype. These are eliminated and the intermediate phenotype is favored. So after selection we get this type of the graph. Third is disruptive or diversifying selection. Extreme phenotypes are favored and intermediate ones are not favored. So these extreme phenotypes have advantage over the intermediate ones. Homogeneous population is broken into several adaptive forms. Phenotype of populations are pushed from its mean value. It tends to increase the amount of variance within the populations because now the population is no more homogeneous. It gives rise to a bi or trimodal distribution in adults instead of unimodal as earlier. It is less common than the directional and stabilizing selection. Salient features: breaks homologous population into several different adaptive forms. Extreme phenotypes have highest fitness. Intermediate phenotypes or mean value have the lowest fitness, that is, are relatively disadvantageous. If we see this graph, this is before The shaded region shows the intermediate phenotype or the mean value, and these are the extreme phenotypes. So these extreme phenotypes are favored and intermediate ones are not favored, and hence the population breaks into two adaptive forms. So instead of unimodal, it is now bimodal. So now let us compare the three types of the selection. This is the population before selection. Frequency is in y-axis and phenotype is in x-axis. This is the mean. In the directional selection, before the selection, the population is like this, which is shown by dotted lines, and this is after the selection. So it moves in one direction. Due to the change in the environment in one direction, individuals are eliminated from the one end and these are added towards the other end. Second is the stabilizing selection. The dotted lines show the graph before the selection and this is the graph after the selection. It takes place in the unchanging environment. Here the extreme phenotypes are eliminated and intermediate ones are favored. This is the disruptive selection, the dotted lines before the selection, and this is after the selection. Extreme phenotypes are favored and intermediate phenotypes are not favored, and hence the population, which is homogeneous earlier, breaks into two adaptive forms. We are going to discuss about evidences of natural selection or role of natural selection. First is Lederberg's replica plating experiment. It was conducted by Joshua Lederberg and Esther Lederberg in E. coli, which is bacteria. Bacterial cells were first cultured on agar plate, and on these agar plate many colonies developed. Then velvet block was pressed against the surface of culture. Cells from each colonies were picked by velvet block. This block was then pressed against plate containing penicillin. Most cells failed to grow on penicillin medium, but few were able to grow. So what it was concluded that these cells which are able to grow on penicillin medium are resistant to penicillin. But how? Drug penicillin itself does not induce resistance. It was proved by taking cells from the original colonies that were never exposed to penicillin. Then these were tested for resistance to penicillin and it was found that these are resistant. So penicillin simply selects against susceptible bacteria by killing them. Or in other words, penicillin selects the resistant bacteria and kills those that are susceptible or sensitive. There are certain genes that are responsible for the resistance, and these develop due to the earlier random mutation. These are already present in the population. Earlier these are present in the low frequency because these do not have advantage over the one which consists of the normal genes and are susceptible to penicillin because these were grown on the normal agar medium. But when these are grown on the penicillin medium, then these have the advantage over the normal ones. Hence those which possess these mutated genes are pre-adapted to survive penicillin treatment. That is, these are already adapted to survive the penicillin treatment. It is not that these develop the resistance due to the exposure to the penicillin. In the presence of the penicillin, these reproduce and perpetuate. Therefore, next generation shows marked resistance to penicillin because in the next generation there are more resistant bacteria because these have the advantage over the susceptible ones. And continued selection, or when these are continuously grown on the penicillin medium generation after generation, gradually increased resistance. That is, there is gradual increase in the resistant bacteria in the population. Therefore, what we can say that natural selection has selected and supported the progeny carrying the beneficial mutation. And this results in the resistant bacteria to evolve, or hence evolved resistant bacterial population. Let us understand this Lederberg's replica plating experiment with this figure. This is the master plate, that is the agar containing medium on which the bacterial cells, that is E. coli, is grown. Then the sterile velvet is pressed against this. It gives imprints of all colonies. Then this is pressed against the two fresh media, one which lacks penicillin and other which contains penicillin. Then these are incubated. So in this medium all the colonies grow. Here only two colonies are growing. These are the penicillin resistance colonies which are able to grow in the presence of the penicillin. Others are not grown if we compare it to the master plate. So we can say that the other colonies which do not grow on this penicillin containing plate are actually the penicillin sensitive cells. So if we compare these two plates with this master plate, we can tell these three colonies. These are the penicillin sensitive and these are penicillin resistance. These are already present. It is not that penicillin's resistance develop when it is exposed to penicillin. It is only that natural selection, in this case penicillin, selects the penicillin resistant cells over the penicillin sensitive ones. Now the second is sickle cell anemia with relation to malaria. Sickle cell anemia we have already discussed in genetics. You can refer my video, the link is given in the description box. This sickle cell anemia is caused by recessive gene. And this gene is responsible for the production of abnormal hemoglobin. And the red blood cells which have this type of the hemoglobin are distorted or sickle shaped. Hence the name sickle cell anemia. Homozygous individuals which have two copies of this recessive gene die early in life due to severe anemia. But heterozygous individual which has only one copy of the gene have mild anemia. Also, these have one advantage. These are naturally protected against contracting malaria. But how? Because these heterozygous individuals have both normal and abnormal RBC. Parasite cannot live in these abnormal RBC which is distorted or sickle shaped. And so they are protected against malaria. Hence heterozygotes survive better in malaria endemic region. Endemic means disease or condition that is regularly found among particular people or in certain area. In these regions, population have both normal and heterozygous individuals. These heterozygous individuals act as carriers of gene for sickle cell anemia from generation to generation, and these are recessive gene which are responsible for sickle cell anemia. It is entirely disadvantageous in one environment and advantageous or beneficial in another environment. That is the environment where malaria is endemic. Hence can be selected in these regions or environment. This explains high proportion of this gene in geographical areas where malaria is endemic. Now third is malaria in relation to glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase is an enzyme and its deficiency is X-linked hereditary disease. In glucose metabolism, conversion of reduced NADPH to oxidized NADP is catalyzed by glucose 6-phosphate dehydrogenase enzyme. Also, glucose 6-phosphate dehydrogenase deficiency is characterized by low levels of reduced glutathione in RBCs. Reduced glutathione level is maintained by glutathione reductase and its coenzyme NADPH. And we have seen earlier that this glucose 6-phosphate dehydrogenase is required for the adequate supply of NADPH. And this enzyme is encoded by the gene that is present in the X chromosome, which is sex chromosome. Glucose 6-phosphate dehydrogenase deficiency leads to hemolytic anemia and prolonged neonatal jaundice. Hence, if there is hemolysis of RBCs, therefore malarial parasites are unable to multiply in these RBCs because malarial parasite breed on these RBCs. Therefore, decreased glucose 6-phosphate dehydrogenase activity within their RBCs leads to resistance to malarial fever. This is beneficial in malaria infested regions and is favored by natural selection. Example, 50% population of Negroes are deficient in glucose 6-phosphate dehydrogenase because these are selected over the normal ones, as these are inhabitants of malaria infested regions. And this glucose 6-phosphate dehydrogenase deficiency is caused by the recessive gene present on the X chromosome. So males are more affected because these have XY sex chromosome, and hence there is only one X chromosome. So one copy of gene is responsible for this disease. Now fourth is industrial melanism. There are two varieties of peppered moth. Scientific name Biston betullaria. One is light colored, which is known as non-melanic variety, and the other is dark colored, known as melanic variety. Now we will discuss about two scenarios. First is before industrial revolution. Peppered moths used to rest on tree trunks and rocks. So these tree trunks and rocks were covered with light colored lichens. Hence, against this light colored background, light individuals blended, but dark moths were conspicuous and easy prey for birds. As a result of which, dark colored moths were very rare because light colored moths survive, and most of the dark colored moths were eaten by the birds. Hence, these were not able to reproduce and produce their offsprings. Now, the second scenario. During Industrial Revolution in Europe, that was in 19th century, industrial pollution soot darkened the tree trunk and rocks because these soots covered their surface. Against this darkened background, now dark colored moths blended and light moths were easily visible to the predators. So frequency of dark colored moth in the population started increasing, that is favored reproductively, but most of the light colored moth were preyed, and hence these were not reproductively favored. So what we can say that natural selection, in this case it is predation by birds, edited the population. That is, it resulted in the shift in the composition of Biston betullaria population in industrial regions. That is from light colored moth to dark colored moth. Now let us understand industrial melanism with this figure. This is the earlier situation where there is no industrial revolution. So when the moth rests on the tree trunk, light colored moth easily blend with the color of the tree trunk or the surface, but the dark colored moths are easily visible, so these are eaten by the predators, and hence their percentage of frequency is less in the population. So we can say light colored moth is greater in percentage and dark colored moth are lesser in the percentage. Later during the Industrial Revolution, due to the soot that come from the industries, these cover the surface of the tree trunk. So now light colored moths are conspicuous but the dark colored moth blends. As a result of which these are easily eaten by the predators compared to the dark colored moth. Hence now in the population dark colored moth are more in number of percentage compared to the light colored moth. So with the Industrial Revolution, the population of moth shifted from light colored moth in greater percentage to dark colored moth in greater percentage or frequency in the population. We are going to discuss about limitations of natural selection. So we all know that main aim of natural selection is to drive populations towards improvement in order to face environmental challenges, because we know that environment keeps on changing. So for the better adaptation of the population or the organism, so they can survive in the changing environment, natural selection plays an important role, and the better adapted individual then reproduce and produces the offsprings of its own kind. This is known as differential reproduction, which we have discussed earlier. But it is not that natural selection is completely perfect. It has some limitations. Let us see these limitations one by one. It operates on genetic variations which is already present in population. It cannot create new variations on the basis of the requirement of the environmental change, so that the population can cope with the environmental change and get better adapted. Certain variations are required, but this natural selection can function only on the variation that is already present. It cannot create the new variations. To proceed evolution in required direction, alleles for certain variation should be already present from mutations and gene flow, because as we have discussed earlier, it cannot be created. So natural selection has to work on what is already present. Selection leads to slight modification within a population over a limited time span. Over the short period of time, only the slight modification takes place in the population. And if there is drastic changes, it may occur by the slow accumulation of lesser changes over many generations in the course of long time, that is millions of years. So for the drastic changes, very long time is required. In case of mutations with large effect, they poorly coordinate with the rest of the evolving organisms, so deviate from normal and are eliminated by stabilizing selection. If by the mutation some drastic change occurs, that is the large effect, then since all the other organisms are evolving at the slow pace, so this cannot coordinate with these organisms, and hence these deviate from the normal and are eliminated by the stabilizing selection, because stabilizing selection prefer the normal or the mean phenotype and extreme phenotypes are eliminated. Sometimes one feature changed for the better brings another change for the worse. Therefore, the result is no net increase in fitness. If any feature change and makes them better adapted, then certain another changes takes place which is not beneficial for the organism. So if we see the net result, it is 0. That is, there is no net increase in the fitness. because on one side it is becoming better and the other side it is becoming worse. And how this is possible? Because an allele that produce a particular adaptive feature has other effect also, and that may include non-adaptive ones. So what is the outcome? A compromise in design rather than perfection. Example, male peacock's large and brilliantly colored tail. So male has large and brilliantly colored tail in order to attract its mate, that is female. But on the other hand, these large and beautiful feathers is hindrance, because these are easily attacked by the predators. These are preyed easily because these are spotted easily, and because of their large feathers these cannot protect them easily, and hence there is a compromise in the design. One side it is attracted by the female easily, on the other side it is harmful because these cannot protect themselves from the predators. Similarly, the ancestors of human beings, they were four-footed, but now in the course of evolution these are evolved into two-footed. This leads to certain problems like the low back pain and also difficulty in childbirth. Also, harmful alleles can be introduced and helpful alleles can be erased. This happens due to the migration and due to the natural disasters, which leads to gene flow and genetic drift. So sometimes adaptations fail, which causes extinction of species during environmental changes. Because sometimes what happens, environmental change takes place at the fast rate, but organisms cannot cope with these changes because these keep changing at a slow pace. Hence, sometimes these become extinct because these cannot cope with the environmental change. Many species that ever lived became extinct in this manner. One of the important examples is of dinosaur. So, seeing these points, what we can tell that natural selection has limited ability and cannot produce adequate adaptations always. What is the conclusion? Natural selection does not lead to perfectly adapted organisms. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.